Gene expression is used by all known life. From single-cell bacteria to the blue whale, every living or once living organism is composed of cells that store the complete genetic information to assemble macromolecular proteins for the organism's biological processes. Located in the nucleus of cells, genes are comprised of 4 base pairs. The order and sequence of the base pairs create the unique genetic code that acts as the blueprint for creating new cells. The complete genetic profile is held on the DNA strands. Each DNA contains 50,000 to 100,000 genes, each comprised of hundreds of thousands of base pairs. This is a lot of information, but not all of the information is used. It is gene expression that determines which portion of the genome is active, and thus dictates which cell or molecule is produced. Through complex regulation mechanisms, gene expression is controlled by signals from the environment as well as from other cells. RNA sequencing technology enables gene expression to be quantified, allowing connections between genes and the environmental stimuli to be made. Let us examine the procedure for determining the gene expression for virus response in the lung cells of a mouse.
To begin RNA sequencing, mice are sedated. A special strain of the flu, called a murine-adapted influenza, has been created to mimic human symptoms in the mouse model. A specified concentration of this virus, called the viral titer or viral load, is suspended in an aerosol inoculum and administered to the unconscious mouse through its airways. We wait some time for the infection to progress and the immune system to respond before euthanizing the mouse for sampling. Normally, mice are sampled at intervals of 0, 3, 5, and 7 days. The organ of interest, in this case the lungs, are carefully removed and either flash frozen for later processing or placed into a solvent and homogenized immediately. The RNA is isolated, fragmented, and randomly primed in a series of chemical treatment steps to prepare it for sequencing. The RNA-rich liquid is placed into an RNA sequence analyzer and processed. Once each fragment of RNA has been tallied up in a raw read, fragments are aligned with a mouse reference genome. The number of fragments at each point along this genome is recorded. We can utilize our knowledge of genomics to determine the expression of individual genes. This expression will differ from a healthy mouse. It will display high activity in immune regulation, repair, and inflammation. Studying these differences gives insight into the mechanism and progression of the disease, and can be used to improve existing treatments and find targets for new therapeutics.